Most insects have two main visual organs, called the compound eyes. They are composed of lens-capped units called ommatidia. Underneath the lenses, photoreceptors' light-sensitive parts, called the rhabdomeres, convert light into an electrical signal that goes deeper in the brain. But breakthrough research has showed that photoreceptors do more. When exposed to light flashes, photoreceptors themselves contract and deform without any muscle. In the eye, these contractions cause rapid rhabdomere displacements called photoreceptor microsaccades. They enable the flies to see much finer details than previously believed and perceive depth. This microscope system views retinal movements non-invasively in living flies. The fly is on a pipette tip. And we have two rotation stages. Things get interesting under infrared back illumination. We can use computer simulations to see what is going on. These are the rhabdomere tips. Under infrared illumination they appear bright, but we cannot see them because of the lenses. Real microscopes have a narrow depth of field. Let's focus deeper inside the eye. Bit too much. This is the deep pseudopupil. It is a magnified virtual image of the rhabdomere tips that we saw earlier. And when the rhabdomeres move, the deep pseudopupil follows, allowing us to measure the rhabdomere's movement. We activated the photoreceptors facing towards the microscope with bright light flashes, and by systematically rotating the fly and repeating the flashes, we can map out the photoreceptor microsaccades across the eyes. Now we can see that the microsaccades are mirror symmetric between the left and right eyes, and if you look closely, you can see that they move in opposing directions at different parts of the eye. These directions align with the rhabdomeric patterning. But why? We compared the microsaccades against the optic flow during forward locomotion, while simultaneously varying the fly's rotation along the pitch axis. Many of these rotations can be experienced during the fly's normal flight maneuvers. Some, maybe not. We found out that in a typical flight rotation, the fast phase matches with the ground flow and the opposite slow phase matches with the sky flow. The fast microsaccades likely aid to resolve intricate visual clutter on the ground, whereas the slower microsaccades enhance the perception of the surrounding landscape and sky. We also tested varying the yaw rotation in a similar way. Here the things get interesting. No more are the optic flows experienced by the left and right eyes mirror symmetric, but depending on the rotation, they can be almost mirror opposite. We found out that when taking a turn, one eye is synchronized with the field rotation, while the other's eyes faces exhibit the reverse pattern, helping to enhance the binocular contrast differences in the surrounding environment.